Hey, would you like to hear something amazing? It's all about BT corn. Okay, sorry, that was a corny joke. Well, anyway, welcome back to our series of videos on molecular genetics. So in the last video, we introduced ourselves to the idea of genetic engineering, and we looked at some steps at how genetic engineering could be carried out, and we looked at the case study of transgenic bacteria used to make human insulin. Now in this video, we'll take a look at a few more case studies just to see different applications that people have used genetic engineering for. So another main application of genetic engineering is in the creation of pest-resistant crops. Now farmers grow crops all the time, and one of the biggest problems they face are pests that eat up the crops before they can be harvested. Over here is a corn, which has been eaten up by pests. It looks really ugly. Over here, introducing to you BT corn. BT corn is a result of genetic engineering. It's genetically modified. It's called BT corn because it contains a gene from a bacteria called Bacillus thuringiensis. I'm not going to give away too much because some of you will be researching on this corn, so please tell us more during your presentation. But essentially, here are the steps in making BT corn. So first, again, we isolate the desired gene. In this case, it is the pest resistance gene from that bacterium. Then we insert the gene into the bacterial plasmid, similar to what we did with the human insulin gene. And then we insert the recombinant plasmid back into the bacteria. Again, similar to what we did with the human insulin gene. Now the difference is, in this case, we don't stop with just putting the plasmid into bacteria, because we still want to put that pest-resistant gene into our plant. So what we do is that we allow this bacteria, for example, a bacteria called agrobacterium, to infect plant cells. Yes, we actually let the bacteria infect the plant cells and hence insert their gene into the plant cells. So as a result, the plant cells now contain the pest resistance gene, and we can culture the plant cells and grow them into a transgenic plant. Now, one of the things that we should clarify at this point is, what is the vector in this case? Now remember, the vector is whatever is used to insert the gene into your recipient organism. In this case, your recipient organism is your plant cells. Now when we looked at the example of transgenic bacteria, the vector was the recombinant plasmid, right? Because I put that into my bacteria, which is the recipient organism. In this case, the vector would be the bacteria that infects the plant cell. The vector in this case is not the plasmid, but rather the bacteria, because the bacteria is the one that is used to insert that gene into our recipient organism, which is the plant cell. So here's a very simple diagram. Okay, not so simple. Let me show you what we just talked about in a bit more detail. So just focus here. This black color part here, this represents the pest resistance gene. And so what we've done is that we've cut the pest resistance gene out using restriction enzymes, and we've inserted it into a plasmid. So here's our recombinant plasmid made up of that pest resistance gene and the plasmid. And remember, we use DNA ligase to seal that bond between the pest resistance gene and the plasmid. Now this is a recombinant plasmid, and we insert it back into a bacteria. In this case, we insert it into a bacteria called agrobacterium tumefaciens. Now this bacteria is very special. It is able to insert its plasmid DNA into plant cells. So when we use this bacteria, we infect some plant cells. So here are our plant cells. We put the bacteria inside to infect them, and some of the bacteria will then infect and place their plasmid DNA into the plant cells. So we select the plant cells which have the gene, and then we then grow those plant cells that have the pest resistance gene, and we generate a new plant which is transgenic in nature. Alright, another related case study also has to do with pest resistance. In this case, this is pest resistance wheat. And why this example is helpful is because it helps us see that genes can also be transferred between the same species. In the case of BT corn, genes were transferred between two different species, right? We're transferring from a bacteria to a corn plant. In this case, we are transferring a resistance gene from a wild species of wheat to a common species of wheat which is used in crops. Okay, so we can also take genes from the same species and put it into a sort of closely related species of the plant. So here's the pest, common pest that destroys wheat, and the pest resistance gene helps to resist these pests. Now, one other very interesting and up-and-coming application of genetic engineering is this idea of gene therapy. Now, what gene therapy does is that you take a healthy gene from one person and then you transfer it into the cells of another person who has a defective gene. And why this is very interesting is because people think this might be the way to heal certain diseases which are due to defective genes, due to mutations. So if you remember sickle cell anemia, that was due to a gene mutation. Right? There was a mutation in the gene for hemoglobin. So imagine if I can take a healthy version of that gene and insert it into a person who has a mutated gene. I could actually make that person recover. Right? And this idea of gene editing, gene therapy is fast and growing. Look at this. This is in 2017. Very, very recent. So here's how it might work, gene therapy. Okay, what we can do is we can actually take out some cells, some stem cells from the affected person, from the sick person. And here are the stem cells of that sick person. So these stem cells do not have a healthy gene. What we do then is that we get a vector. In this case, the vector would usually be a virus. And this virus would have the whole hemoglobin gene inside, right? The healthy hemoglobin gene inside. We've inserted the healthy hemoglobin gene into the virus. Then we use the virus, which is the vector. We use the virus to infect these stem cells and to insert the healthy gene into these stem cells. So over here now we have stem cells which have the healthy hemoglobin gene, and then we put these cells back into the patient, and these stem cells, which now have the healthy hemoglobin gene, are able to produce red blood cells with healthy hemoglobin. So we hope that through such technology, we'll be able to help people who have genetic diseases. Now, just a final note before we move on, that there are actually other methods of genetic modification. So what we have seen so far is genetic engineering involving the transferring of genes, right? We've seen genes being transferred from one organism to another organism. That's genetic engineering. But there are also other ways you can modify an organism's genes. You could delete some genes from that organism, or what some people do is you could even silence those genes. That means those genes are still there, but you make tweaks so that they are not expressed. So I'm just saying this because well, there are some people who use the term genetic modification and genetic engineering interchangeably. So they use it as though it meant the same thing. But some people would use it differently. So they will say that genetic modification is more general because you could modify by transferring genes, you can modify by deleting genes, you can modify by silencing genes. Whereas genetic engineering refers specifically to transferring genes. Okay, so the last thing I'm going to cover is the difference. It's a comparison between genetic engineering and selective breeding. So remember when we looked at artificial selection, we looked at this idea of selective breeding, and we looked at this idea of hybridization. So for example, I can take a sugar cane with good amounts of sugar, and I cross it with a sugar cane that is resistant to pests, maybe. I cross these two together and eventually I can get a sugar cane with both good traits. Right, so that's hybridization. So getting a hybrid of two individuals with desirable traits. So what's the difference between genetic engineering and selective breeding? Well, the first thing is, selective breeding, you can only transfer genes between closely related organisms or organisms of the same species. Right? Because if they are very, very different species, you just can't breed them. you can't make them breed with one another. Right? I wouldn't be able to breed a bacteria with a human being. That would be strange. However, in genetic engineering, you can actually technically transfer genes between any species, even between non-related species, right? We've seen that. I'm transferring genes from bacteria to corn. That's crazy. Now, in selective breeding, you might also end up passing down defective genes to the offspring because we're just taking two parents and breeding them together, right? So we want the good traits from the parents, but the bad traits, the defective genes, may also be passed down to the offspring. We have no control of that. Whereas in genetic engineering, we have isolated that desired gene. So we only take out that desired gene and we only transfer that desired gene. So you don't get any defective genes being transferred. Next, selective breeding is much slower, right? You've got to get the two parents to breed together, you've got to wait for the offspring to grow. It's going to take several generations. But in genetic engineering, it's a very rapid process. Right? I can modify an individual cell, and then after that I can cause these cells to reproduce very quickly. So in the case of that BT corn, I modify a few plant cells, and then I get those plant cells to divide, and to grow into a new plant. So it's a much faster process than breeding. So in the same way, selective breeding is less efficient, while genetic engineering is a lot more efficient, as we can grow those transgenic organisms much faster and they don't require as much support in terms of food. Alright, so that's the end of this video, and in the next lesson, we're going to take a look together at what are the social and ethical implications of genetic engineering. I mean, it sounds really, really cool, but what concerns could come up? What kinds of issues might crop up? Well, it's up to you guys to share that during your presentation in class.